equi, giusto?
Quindi 4 per 2 è 8. 4 per 2 è 8. E
topical therapy for allergic dogs. We are talking about antimicrobial, antipruritic, anti-inflammatory topical therapy. Okay, so this is everything we're going to see tonight. We're going to see, first of all, anti-inflammatory and antipruritic topical therapy, and then barrier and microbiome restorers, disinfectants, antiseptics, essential oils, antimicrobial peptides, and biofilm dissolving agents. So first of all, we are going to see what is now new in anti-inflammatory and antipruritic topical therapy. And we're talking about animals with atopic dermatitis. First of all, there is a whole new concept regarding how to treat allergic animals. Until now, we have treated them with a reactive way. So whenever the animal has pruritus, has inflammation, we treat until the pruritus and the inflammation goes away, and then we stopped the therapy and we would wait that the pruritus and the inflammation come back, and then we treat again. So this is a way to run after the inflammation. But in fact, now the new guidelines in human medicine and also in veterinary medicine, they say that we have to prevent the relapse. We have to treat constantly in order to avoid the returning of the inflammation and of pruritus. And this is called a proactive therapy. So we can treat once to have the inflammation and the pruritus gone, but then we never stop treating. We continue with something that is, of course, not as strong, and it must be very safe because maybe you have to give it for many months or for the whole life, but you continue giving something that will prevent the relapse of the clinical signs. And one of the products that has been evaluated as a proactive topical treatment is Cortavans, hydrocortisone aceponate. So it has been used in order to avoid the relapse, maybe once a day or twice a week on the skin area where we would expect to have the relapse of pruritus and of erythema. And it has also been seen that when we treat as a proactive way with Cortavans or with any other anti-inflammatory topical therapy, we're going to be able to keep the microbiome in balance, because all inflammation is leading to a dysbiosis, but if we inhibit inflammation, we are going to improve the microbiome and to avoid the dysbiosis. And if we want to use a corticosteroid in order to decrease the inflammation for a long period of time topically, we must use some topical therapy that is not thinning the skin and is not absorbed through the skin, because maybe we give it for months or for years. And hydrocortisone aceponate is one of these corticosteroids that is not causing skin thinning and is not absorbed systemically. These are called soft steroids or smart steroids. Another one is, for example, mometazone furoate. So this study with Cortavans has compared 41 dogs that they had a mild atopic dermatitis in remission, and half of the dogs were treated with Cortavans and half of the dogs were treated with placebo on 2 consecutive days as a proactive therapy, on the weekend therapy, so Saturday and Sunday, in order to avoid the relapse of atopic dermatitis. And now the dogs that were treated with the active ingredient, they had a relapse after an average of 115 days, and some dog nearly after 1 year, while the dogs that were treated with the placebo, they had a relapse after 1 month with a range of 15 to 61 days. So this is a very nice strategy to prevent the relapse of the clinical signs of atopic dermatitis without using systemic drugs. This was done on the skin, but you can do the same if you want to prevent the relapse of the otitis in allergic animals. So they did the same thing. They just sprayed Cortavans in the ears, also in the weekend, in order to prevent the relapse of the otitis after they had treated the otitis and they had brought the otitis under control. But then, when the relapse of the otitis, sprayed Cortavans. Again, they have done a group that was treated and a group that was placebo. The placebo group had a relapse after 90 days and the treated group had a relapse after 180 days as an average. But the number also of dogs that had the relapse with the Cortavans was very, very low, only 5% versus 50% of dogs that was treated with a placebo. And this is another very similar study, daily Cortavans for a couple of weeks and then again on the weekend versus placebo. And here we have a very similar result. Relapse rate of dogs that were treated with placebo, 50%. Relapse rate of dogs that were treated with Cortavans, much lower. And so this was in the ears. Okay, so in this case, in order to decrease the inflammation, we actually use a drug. But we can decrease the inflammation and improve the health of the skin of allergic animals by using other means, by using barrier restorers or microbiome restorers, because we know that in animals that have atopic dermatitis, the defensive lipid skin barrier is defective, and also there is a dysbiosis of the microbiome. And these two factors, they will contribute very much for the inflammation, for the pruritus, for the entrance of allergens and microbes. So how is this lipid barrier made? Here we have the epidermis, here we have the stratum corneum, the corneocytes, and this black material is the lipid material that is between the corneocytes. And these lipids are long-chain ceramides, long-chain fatty acids, and small cholesterol molecules. And here you see the percentages. The majority is ceramides, very important for a good, effective, defensive lipid barrier. And then we have free fatty acids and cholesterol. And here you see how they are all packed in a very orderly, organized way, so that they make some leaflets, some layers that are nicely put between the cells, and they will seal the cell very well, so that they will provide water resistance. These are lipids, so the water cannot get out of the skin, and so the skin will remain well hydrated. If you have a bad lipid barrier, you will have a lot of transepidermal water loss, and you will have a very dry skin. And measuring the transepidermal water loss, there are some machines that are able to measure how much water comes out of the skin, is actually a way of measuring if your lipid barrier is intact or is defective. And then it will also defend from anything that comes from outside, particularly if these things are water soluble. I show you here, this is a healthy animal. This is an electron microscopy photo. This is a keratinocyte, and this is another keratinocyte. And here it is the space between two keratinocytes. And you can see here all these layers that are all very nicely in order. This is a healthy animal, and these are all the lipid layers that are sealing the space between the two keratinocytes. This is an atopic dog. You see that there is no order, there is a mess, everything is disorganized, and what happens really is that there is a lack of ceramides, and there is too much cholesterol, and the lipids are not able to organize well. You understand that this situation is not offering the sealing protection of a normal intercellular lipid barrier. So what can we do for that? Well, we can use topical products that are able to help restore the lipid barrier. And there are many in the veterinary medicine that are on the market. You will recognize here from the ICF Hermidra. We're going to talk about that later. And you can have also different formulations like spot-on, sprays, and so on. And in the human medicine, there is even liquid ceramides. So they are just putting what is missing, the missing ceramides in the atopic skin. So I talked about studies that have been published, and there are many, many studies with a variety of products, because these products are very popular. And so I'm just putting all the titles of the studies that I have found that are talking about the topical fatty acids and the topical ceramides and the topical sprays to restore the Statrum corneum. I would like to talk to you about a study, and that is a study made with the product Hermidra that I mentioned before, which is of ICF. That is a spray, and Hermidra has a variety of active ingredients. First of all, these active ingredients, they are encapsulated in liposomes. And these liposomes contain these ingredients, and they are like onions, microspheres made of many lamellae. And each time that the outer part of the lamella is lost, then some active ingredient is released on the skin of the dog. And then one more sheet of the onion is lost, and then you release more product. And this makes it possible that all these active ingredients are actually released very slowly and very gradually. Let us see what are these ingredients. One is zinc. You know, zinc is keratoplastic. There is also shampoo, Zincoseb, that is for seborrhea. And so it's very good because it will help the formation of a good keratin. And then we have other moisturizing, soothing, softening products like willow herb, which is also astringent. That means that it will regulate the sebum. Pantothenol, which is moisturizer. Propynyl glycol, this is a mineral oil. It is also a good moisturizer and also keratinolytic. And also glycerin, it is also moisturizer and softening. So you're going to add all oils, the lipids that are lacking, but you're also adding some molecules that are going to trap the water inside the skin, because these are moisturizers. So they will trap the water in the epidermis. So when you have a high transepidermal water loss due to your allergy, these products will actually normalize this transepidermal water loss. And this is a study that has been done with Hermidra. And in this study, actually, the animals were completely healthy. These are experimental beagle dogs. And the skin damage was realized was caused artificially. They have taken Scotch test, or sticky tape, and they have put the sticky tape, the Scotch, several types on the same area of the skin. This is called tape stripping. And if you put this sticky tape always on the same area, you're going to take away some layers of your epidermal barrier, because you're going to strip it out. And so you're going to cause a damage on the barrier with tape stripping. And if you have a damage on the barrier of these normal dogs, you're going to have an increased transepidermal water loss. So this damage was caused artificially on the skin, and the transepidermal water loss was quite high. That means the skin barrier was damaged. And in half of these dogs, the damaged skin was treated with Hermidra, and in half of the dogs, the damaged skin was treated with a placebo. When you treat the skin with whatever you put, you will improve your transepidermal water loss, because of course you put something that is liquid, and so it is water-based on the skin. But then the animals that had the placebo, there was no permanent improvement of the transepidermal water loss, because then the damage got back to high transepidermal water loss, high damage as before, while the dogs that received Hermidra, the transepidermal water loss went down and remained down for many hours. That means that the defect was repaired and was really normalized for a long period of time. So in this case, you can really understand how Hermidra is able to improve the transepidermal water loss, which is a sign of barrier defect for a long period of time. Okay, now a defective skin barrier favors, of course, the adhesion and the multiplication of the microorganisms, because if they find a way to infect the skin because the skin barrier is defective, for them it will be able to enter in the skin and cause an infection. So usually the skin of animals with atopic dermatitis has higher amounts of staphylococci if you compare it with the skin of healthy animals. But not only, the severity of the lesions correlate with the amount of the staphylococci. That means that the worse is your erythema, the worse is your inflammation on the skin, and the higher is the number of staphylococci that are adhering in that skin. So there is a direct correlation between atopic lesions and number of staphylococci. The problem is that usually in a healthy skin, you have a high variety of bacteria. You have many different types, not only staphylococci. And I show you, these are healthy animals. Every bar is a different animal. Every bar has different colors, and every color is a different bacterium. So you see how many colors we have here. So normal skin has a lot of different bacteria, and this is healthy. This is how it should be, because all of these bacteria, they live together and they actually fight against the pathogens, because these are all the good resident bacteria. Let us see the skin of dogs that have atopic dermatitis. First of all, you see much less variety of colors. Half of it, or the majority of it, is pink. And pink, of course, is staphylococci. So this means that we have much less species types, much less variety, and much more staphylococci if you compare it to healthy animals. And this, of course, is not good, because Staphylococcus is able to cause pyoderma. And not only, Staphylococcus is able to cause inflammation, pruritus, stimulation through superantigens of the immune system. So what do we want? We want to go back to the normal situation where we have a lot of variety and healthy microbiome. We do not like this dysbiosis situation. Well, we have some new, innovative, interesting, alternative way to reverse this situation. Instead of giving antibiotics and killing all the bacteria, which is also not very good, we would like to reverse the dysbiosis to a normalization of the microbiome of the skin. And one way to do it is to spray good bacteria on the skin. This Link Skin by ICF is a spray that contains lactobacilli, Lactobacillus rhamnosus and reuteri. They are tyndalized. That means that they are dead, but their antigens have not been destroyed. And this is important because these antigens, they interact with the immune system, and they will teach the immune system to kill the staphylococci and to help all the other bacterial species, the good resident ones, to proliferate and kill the staphylococci. And so in this way, you can really, thanks to the help of the immune system, and also thanks to other substances that will help the proliferation of the resident bacteria, you can counteract this dysbiosis and alteration of microbiota. We're going to talk about Link Skin in the second lecture about microbiome. Okay, now we have talked about the fact that sometimes there are many, many bacteria on the skin. You may even have infections. And so you may want to use really disinfectants and antiseptics because these bacteria are too much and they have given an infection. So let us see how big is the problem. So these are all the cases of secondary pyoderma that I have seen in my caseload. These were 1,000 dogs with dermatological diseases. Well, of all the cases with secondary pyoderma, 1/4 had atopic dermatitis, and about 15% had food allergy and 15% had flea bite allergy. Altogether, the animals that have secondary pyoderma, half of the cases, they have an underlying allergy. This means that the association between allergy and bacteria and pyoderma is really very strong. So each time when we see a pyoderma, like in this case, for example, we should always think to look for an underlying allergy. And each time that you have an allergic animal, you should always check if this animal doesn't have a bacteria folliculitis or a bacterial overgrowth or a dysbiosis. This is a bacterial folliculitis, which is so frequent in allergic dogs. It is a follicular disease, so the hairs are lost. And you can see here the patchy alopecia on the dorsum. And when you turn the dog on the other side, you see the collarettes. And here, if you make an impression smear, from the borders of the collarettes, you have pus. And in the pus, you will see, of course, the neutrophils, but you will also see the bacteria. These are these round cocci inside the neutrophils. And the neutrophils will have a swollen nucleus. They are degenerate. They are suffering the fact that there are bacteria in the cytoplasm producing toxins. So in this case, you really have a pyoderma, and you may want to treat it, and you may want to use something that is not an antibiotic, because all the guidelines on the treatment of bacterial skin infection say that because this is a superficial infection, before using an antibiotic, you should consider absolutely topical therapy, topical disinfectants. And we have a very nice Italian study made by my colleagues, Stefano Borio and Silvia Colombo, and they have used chlorhexidine 4% shampoo and spray in dogs that had bacterial infection. Actually, they have taken these dogs with pyoderma and they divided them in two groups. One group used twice weekly chlorhexidine 4% shampoo, chlorhexiderm, and then the days that they were not shampooing the dogs, they would spray with chlorhexidine spray. And the other group was treated with antibiotics orally, amoxicillin clavulanic acid. The results of the two groups are the same. You see that they improve in the same speed and in the same way, the same measure. The only difference is that with the shampoo, they could kill also the amoxicillin resistant bacteria, the methicillin resistant bacteria, so that the shampoo has the advantage that it will kill every bacteria, while the antibiotics only will kill the bacteria to which that are sensitive to this antibiotic. Now, chlorhexidine certainly is probably the best disinfectant that we can use, that we know now. It also has a very long residual effect. But we should not think that we only have chlorhexidine shampoo or spray. We can have chlorhexidine also in other formulations. I use quite a lot these wipes, a lot, for example, for skin folds, interdigital spaces, skin folds on the muzzle that are secondarily infected. And here you see this is a study on the residual activity of chlorhexidine, and in CLX wipes, which is not CHUX but is CLH wipes, they have found a long residual effect, much better than with other wipes. for methicillin-sensitive Staphylococcus, methicillin-resistant Staphylococcus, no difference, resistant to sensitive. Chlorhexidine will kill all the bacteria, and it will also kill Malassezia pachydermatis, also a long residual time for the treatment of Malassezia. So if we have to summarize, the good things of chlorhexidine is that the efficacy, it has been seen in many studies, is superior to other disinfectants and is the one that has the longest residual activity, even after rinsing if you have a shampoo. However, you have to remember that now we start maybe seeing some reports of resistance to chlorhexidine. So we hope to be able to use it also in the future. We will see if these reports will be confirmed or not. Now, what would be an alternative to chlorhexidine? Well, there are some studies that have evaluated sodium hypochlorite. This is the house bleach, the one that you have in your houses to disinfect and to bleach. It is used in human medicine, for example, in atopic children that have dysbiosis with staphylococci on their skin. They make whole body baths with some sodium hypochlorite. In these children, they improve very much because you give quite a good disinfection to the skin. And this has also been studied in dogs. And it has been seen that at this dilution, 0.05 to 0.005%, it does not alter the barrier integrity and it does not kill the keratinocytes. However, the problem is that the action is not so long. This is the bleach treated areas, and you see that the number of bacteria goes down, but not so quickly and not for a long time. It then will go up again quite quickly. And this is the water treated areas, and there is not a lot of difference in the results between the two areas. So I'm not sure that this will be a good option for the future. The advantage is that it is quite cheap. If you want to try it for a homemade disinfection, which is not very toxic, if you can dilute in 10 liters water, 30 milliliters of 1.5% bleach, or 10 milliliters of the more concentrated bleach, and 1 tablespoon is more or less 10 milliliters. Okay, let us see some alternatives for the disinfection. And I would like now to talk about essential oils, because they have been considered by many studies recently, and we have more and more publications on essential oils. What are essential oils? They are mixtures of volatile components, mostly terpenes, phenylpropanoids, and coumarins. And we know more than 500 active components from vegetables, and they are extracted from flowers, leaves, wood, roots, seeds, and bark distillation. These are interesting because these are molecules that are produced by these vegetables in order to defend themselves against bacteria and fungi and viruses. So these molecules are actually produced by the vegetables as disinfectant to kill their microorganisms and germs. And so we can use them also to kill microorganisms. So we have studies in vitro. For example, you may have heard about Manuka. So we have here in vitro study against Staphylococcus pseudointermedius. You may have heard about tea tree, jojoba, avocado. Again, we have this mixture against Staphylococcus and Malassezia. Rosemary, this is really big in Italy because we have Mediterranean vegetation with a lot of rosemary, and again, Staphylococcus, everything in vitro. We don't have a lot of studies in vivo with these vegetable oils, essential oils. We only have studies with Manuka oil, for example, and we have studies with a product that is called BioSpray. And this PO spray based on vegetable oils has been used in dogs that had pyoderma. However, it has been evaluated associated to an antibiotic. And the dogs that had the vegetable oil spray and the antibiotic together, their healing was quicker compared to the dogs that were treated only with the antibiotic. Okay, I am very happy that it will increase the rapidity with which my antibiotic works, but I don't want to give antibiotics at all. I would like to eliminate the antibiotics. I would only like to give topical therapies. So there is one more study with the same product that has evaluated topical therapy without antibiotics. So the efficacy of this product with vegetable oils on superficial pyoderma alone. And here you see that the lesions after 10 days will improve by 23%, and after 3 weeks, they will be improved by 37%. I am happy that they improve, but in my opinion, this improvement is not enough. I would have liked to see a much bigger improvement. This was the extension of the lesions. The severity of the lesion also has a better improvement. Severity of the lesion after 3 weeks is improved by 61%, which is interesting, but I'm not sure that it would be satisfactory in every case. We also have some problems with these essential oils, that they are very natural products, so they derive from plants, and so their quality really depends a lot from the batch, from batch to batch. It will depend on the vegetable part that you take, the age of the plant, the vegetative state, the soil composition, even on the climate. Here, for example, you have a study, this one, that has analyzed the content of some active ingredients. Here you see the names of the active ingredients in essential oils of Artemisia in different batches. And you can see the big differences for each one of these ingredients, from 20 to 76%, from traces nothing to 36%. So you see the variability is really big. So it will be difficult to have a really standardized product because it's really natural. Another interesting concept, in my opinion, is that the plant's extracts are able to stimulate the production of antimicrobic peptides by the keratinocytes. This has been proven with Boldo from Peru and Olmare from Eurasia in vitro. So they have put these vegetable extracts in keratinocyte cultures, and they have seen that these keratinocytes were producing more antimicrobial peptides. This is called the defensins technology, and this has been incorporated in products by Virbac. And this has been also studied in vivo. So there are these extracts that in contact with the keratinocytes will stimulate the keratinocytes to produce some products that will kill the bacteria. And here you have the placebo, and this is the number of bacteria, day 0, day 14, day 28. And here you have the products with the defensin technology. And you see that there is a decrease in the number of bacteria by 76% within the first 2 weeks. Why this goes up again after another 2 weeks, I do not know. But the concept of using antimicrobial peptides to kill the bacteria is really interesting. And so I'm going to talk a little bit about antimicrobial peptides. So these are very small molecules that are produced by all living beings, by plants, by bacteria, fungi, animals, humans. And we know they are many, 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 and they have more than 3,000 antimicrobial peptides that have different properties. They can be antibacterial, antifungal, antiviral, antiparasitic, antitumoral, or immune modulating. And these are molecules that we have been knowing for many, many years. For example, you know the old antibiotics, polymyxin A and colistin, they are antimicrobial peptides. And they are used as antibiotics. So what are they in fact? Antimicrobial peptides are very small molecules and they can be linear in structure or cyclical. They can be hydrophilic or lipophilic. The important thing is that they always have a positive charge because they will bind to bacteria, because the bacteria have a negative charge. In fact, the cell membranes of humans, mammals, animals, they are neutral. So these antimicrobial peptides will not damage the animal cells. They will only bind to the bacterial cells and they will damage the bacterial cells. How do they damage the bacterial membranes? So they bind to the cell membrane and then they organize in a fashion that they will actually form a hole in the cell membrane of the bacterium. And you can see this electron microscopy photo. These are bacteria, and here you see that there are holes in the cell membrane of the bacteria. And if you have a hole in the cell membrane, then all your water the water goes in and the content of the cytoplasma goes out and your cell, of course, will die. So this is a very nice, elegant way to kill bacteria. Unfortunately, we already start seeing resistance to some of these antimicrobial peptides because the bacteria will always find a way to develop resistance. So there is also the research and production of artificial antimicrobial peptides. And ICF has searched among many, many different molecules and has selected one that is called AMP 2041. And they selected this molecule because it has shown efficacy on gram-positive bacteria like Staphylococci and Streptococci, but also on gram-negative bacteria like Pseudomonas and coli. And you can recognize that these are the bacteria of our pyoderma and all others are the bacteria of our otitis. Now, they have seen that this antimicrobial peptide, AMP 2041, they can also be potentiated by EDTA. I do not know if you have ever heard that EDTA is actually preparing the bacterial wall, particularly of the gram-negative rods, for the entrance of the antibiotics. So if you pre-treat the bacteria with EDTA, they will die much easier if you use antibiotics afterwards, but also if you use antimicrobial peptides afterwards. And if you put this in association with antibiotics, of course, they will potentiate the action of the antibiotics. Now we have some studies with this AMP 2041, which is included in the product called Peptivet. And these studies are, first of all, in vitro, good results with the dermatological solution, good results with the otological solution, good results with the shampoo, all contain this antimicrobial peptide 2041. And here we have a nice study with the use in vitro of antimicrobial peptide 2041 against Pseudomonas from otitis. And here you see the contact time, and within 5 minutes, over 60% of the Pseudomonas was inactivated. So very, very rapid. And within 30 minutes, 100% of the Pseudomonas was inactivated. Rapid and very potent. So we also have now an in vivo study with the use of Peptivet Auto, the gel. And this was done in Italy. So you see that the title is in Italian, but Slovenia is not so far away from Italy. Maybe some of you can also read Italian. In any case, it's the clinical study on the efficacy and tolerability of this new otological product that contains IMP 2041, Peptivet, in dogs with acute otitis. And the Giovanni Ghibaudo had 36 dogs with bacteria or mixed otitis, bacteria and yeasts. And he did put in the ears every 48 hours this gel. And on day 14, after 2 weeks, he had 60% of the dogs that were completely cured and 11% had a good improvement. So over 70% of the dogs without an antibiotic that were greatly responded to this antimicrobial peptide. Okay, so the next concept I wanted to introduce is the concept of biofilm disrupting agents. First of all, I have to tell you what are biofilms. Well, you have to know that microorganisms, they can live in two different patterns or growing phases. One is the fluctuating planktonic growth. That means these are single cells. They are not attached to each other. And this is what we very often see in our cytology. However, sometimes they are able to adhere on a surface. They start growing on this surface, but then they start producing a mucinous matrix, a polymeric extracellular matrix, and they start replicating in this matrix, and this is called the biofilm. And when they are in this extracellular mucinous sticky material, their behavior is really different. And the behavior is different in that they are less resistant to disinfectants and to antibiotics that come from outside, because they are protected by this mucinous material. And they are also less resistant to antibiotics that come from inside the dog, because in this phase, they are not replicating so much. They are sort of dormant. And usually the antibiotics, they work on the replicating cells. And so the bacteria that do not replicate, they will not be killed and they will resist. Which are the bacteria that are of our dermatological interest that are able to produce biofilm? We know about Pseudomonas, we know about Staphylococcus aureus, Staphylococcus epidermidis, but also Malassezia. So here you have this whitish sticky material in the skin folds, in the muzzle, in the interdigital spaces. And when you do cytology of this material, you will really see a pure culture of bacteria in a sticky mucinous matrix. And here is a recent article where it was also proven that even Malassezia can form a biofilm. Now I'm going to show you what is the increased resistance of the antibiotics. It can be up to 1,000 times because they are protected by this matrix and they are dormant. So in light gray, these are the planktonic normal bacteria, and in black, the biofilm. And these are different antibiotics. And this is the number of bacteria that can be killed by the antibiotics. And you see here, for example, for marbofloxacin, many more bacteria can be killed if they are planktonic, and much less bacteria are killed if they are in the biofilm. And this is the same for this antibiotic. So you see that with the same dosage of antibiotic, much less bacteria can be killed if they are in biofilm. Or the other way around, the minimum inhibitory concentration is much increased if the bacteria are in biofilm. This is Pseudomonas from canine ears, and you see neomycin, the MIC is much, much higher, double so much if the bacteria are in biofilm rather than planktonic, and the same is true for the other antibiotics. So then it would be nice if we could have some drugs that are able to disrupt this biofilm. And some are actually vegetable oils like Manuka oil or even Manuka honey. And here we have some studies on the efficacy in vitro of Manuka oil or honey to disrupt the biofilm formation of Staphylococcus and here of Pseudomonas. When we talk about honey, there is also a nice study that has proven how honey can be useful for the treatment of otitis. Here we have 15 dogs with otitis, 8 with malassezia, 5 with staphylococcus, and 1 with pseudomonas. And they were treated with just medical honey, 1 milliliter per day for 3 weeks. And we have about 70% cure at 14 days and 90% at 21 days. These results at 14 days are quite similar to those we obtained with Peptivet. Also, another treatment that does not include an antibiotic, so we have the advantage that we are not actually causing antibiotic resistance. So are there other products that are able to disrupt biofilm? Well, there is tris-EDTA. We have talked about tris-EDTA before, because tris-EDTA, remember, is able to increase the potency of the antibiotics against bacteria, particularly gram-negative bacteria, because it will increase the permeability of the bacterial membrane to these antibiotics. But not only, it also can increase the efficacy of the antibiotics by disrupting the biofilm. So you see, these are all bacteria that produce biofilm, and this is the minimal inhibitory concentration. And in blue, without tris-EDTA, and in red with tris-EDTA. And you see that for each antibiotic, if you add tris-EDTA, the minimum inhibitory concentration of the antibiotic, which is needed to kill the bacteria, is much lower, thanks to its biofilm disrupting efficacy or properties. And another molecule that is able to disrupt the biofilm is N-acetylcysteine, because it is actually a mucolytic. You know that you take N-acetylcysteine when you have coughing and a cold, because it will dissolve the mucus that you have in your respiratory tract. Well, it has the same mucolytic properties with this mucinous material that we have in the biofilm. So these are two in vitro studies on bacteria that are commonly isolated from otitis externa and otitis media, staphylococci and pseudomonas. And here again, you have the resistant strains. With the biofilm, you have much more resistant strains than bacteria that don't have biofilm. But when you add, hello? But when you add N-acetylcysteine, particularly if you add it at concentrations that are a little bit higher, then you will have no bacteria-resistant strains, not biofilm and not planktonic. Only when you add a very low concentration, then you will still have some resistance. But this is really very low concentration. At higher concentrations, you eliminate all the resistance from the bacteria. Okay, so we have a product that combines tris-EDTA and N-acetylcysteine in one, and this is really used as a biofilm disruptor, and this is Tris-NAC. And it is really nice because when you have this otitis that has a slimy exudate, you will really see that it will clean your ears and eliminate all this slimy exudate. Or in the skin folds, in the perivulval area, sometimes it is really, really helpful. But this is a very nice recent publication. Not only it will disrupt the biofilm in itself, N-acetylcysteine and tris-EDTA, they are also able to kill the bacteria. Here you have the MIC of these two products that are also able on their own to have a disinfectant, antibacterial properties. They seem to be very high MICs, but you have to think that you will actually give high concentrations of these products. So this concentration that we use in this product exceeds by a lot these MIC concentrations that are needed to kill the bacteria. Okay, that was my last slide. I hope that you enjoyed this talk on all that is new in topical therapy, and I am ready to take any questions if you have. Hello? Did you hear my lecture?
Hello, are you there?
I am there. Did you hear
I know, but I'm
everything?
asking the guys.
What?
I'm asking the participants if they are there, but they are there. 37.
Okay, so I did not speak on my own for 1 hour.
No. For sure,
Okay.
I'm here, at least for me. If you have a questions and you don't have it now, there is a chat box on the bottom, so you can write your question and Chiara will answer afterwards.
Yes, or even now, if they want
even
to speak.
now, whatever you want. Please give us a sign that you are here with us.
Okay, everybody now has the possibility to change the microphone on that was first muted. So if you want to, you can ask your question directly.
So any questions? If not, we can then continue.
Okay. Everybody is okay? Or they are...
There are in chat. There are 2 in chat.
Okay. Do you use essential oils, Po? I don't understand the question. Ah, Peros. Okay. No, I have never done so. Although I know that there are people that use the extract from ribes nigrum, for example, as an anti-inflammatory. And now, I don't know if this is an essential oil, but this new cannabis oils are also used as anti-inflammatory, anti-pain products. So not me personally, but I know that there are products on the market.
Some more questions?
Any further questions?
So then continue.
Okay.
Not the time, yeah.
Okay, but I mean, are they enjoying it? Are they alive, or they are all?
I think yes, they are alive.
Dead. Can we have a confirmation from our Slovenian host that everything is okay on their side?
Alenka, are you there?
Hi, Katarina, it's Mojca.
Hi, Mojca. So all is okay?
Everything is fine.
Okay, good. So we continue.
We just have muted microphones so we don't disturb, but we hear you, we see you.
Okay, there is another question.
Continue.
There is another question. I have resistant bacteria on the skin, but also in the subcutis. Do you have any suggestion? If you have bacteria deep in the subcutis, you must use an antibiotic. But if it is a deep infection, you must do a bacteria culture and sensitivity testing in order to choose the best antibiotic, because you have to give it systemically. Okay, so let us go with the second lecture. And the second lecture is a journey suggestion about Zincoseb. What do you mean by suggestion about Zincoseb? It's an anti-seborrheic shampoo. So you use it whenever you have dry or greasy seborrhea, scaling, hyperkeratosis, and so on. I hope I answered. Okay, let us go to the next lecture. So it is a journey in a new world which we have to discover. And in fact, this world, which is so new, so different, we are going to make this nice journey, is inside ourselves. So what is the microbiome? The term microbiome was coined by Joshua Lederberg about 20 years ago, and it defines the ecological community of all commensal, symbiotic, pathogenic microorganisms that are in our body, and with a particular attention on their genome, so their genes and everything they are going to produce as metabolism. And this includes bacteria, archaea, which is something between bacteria and viruses, fungi, protists, so like Leishmania, for example, and arthropods, like Demodex mites that live in our hair follicles, in our eyebrows. Now, in fact, for many years, we knew that there were many more bacteria in our body, for example, in our intestinal tract, than those that we could actually grow in the culture plates. Because I'm sure that it happened to you too, that you have an exudate, for example, from an ear, you see many, many different types of bacteria, and then you send it to the lab and they can only culture 1 or 2 bacteria. And then you say, but I see many more. And they say, but we can only culture a few. Well, over 90% of the bacteria are not able to grow on culture plates. And so until now, we could only know and identify those that were growing on the plates, but we were missing over 90% of the bacteria. So in fact, in 1987, we knew 11 bacterial phyla, and now, after 20, 30 years, we know 35 bacterial phyla, and we know over 3,356,000 different species. This is called the database project, the ribosomal database project. And this is a database that is constantly updated. And when new bacteria are coming, they are added to this project. How are these bacteria recognized? Well, they use PCR. And so every DNA or RNA of the bacteria that are present in a certain sample is amplified, and then it is classified and it is identified. So in order to amplify the RNA from the bacteria, they actually take one gene, which is the 16S ribosomal RNA gene, which is very similar for every bacterium. So that with the PCR, if you check for this gene, you're going to find it and you're going to be able to amplify this gene of every bacteria in the world. And this gene has some variable areas that are very typical for each single bacterial species. So you're not only able to amplify every bacterium in your sample, but you're also able to identify them and differentiate them in different species. And the same can be done with fungi with a gene that is called internal transcribed spacer. So actually from one sample, you generate an incredible amount of data, and you need bioinformatic tools. You need to put all this data in a big computer, and this big computer is going to identify the sequence of these bases, of these genes. They are going to tell you to which class, to which species they belong, and they are also going to make comparisons between the samples. And when you make comparisons, you actually analyze what is the same and what is different between the samples. And so you make this diversity analysis all with the computer, and you can identify 2 types of diversities. One is called alpha diversity and one is called beta diversity. So let us see what is alpha diversity. This is the diversity of this bacteria species within the same sample. So you have one sample and you analyze everything that is inside that sample. For example, the richness, that means how many different species you have. So in this sample, you have 4 different species, and in this sample, you have 2 different species. Or maybe you want to see the evenness, so the relative amount in each different species. For example, in this case, you have more or less the same amount of bacteria for these 4 different species. Here you also have 4 different species, but one has many more bacteria than the other 3. And then you can also identify if these species are related, if they are brothers, cousins, or they are not related. So you can see the phylogenetic relationship between the species. This is the alpha diversity, everything you can say in one sample. Then you have the beta diversity. So you take 2 samples and you compare them. So it can be 2 sites on the same animal or the same sites, axilla and axilla, but on 2 different animals. And you can make the comparisons and then you generate this table and these graphs and you get a lot of information. And now, because this is getting really interesting also for animals and also significant, we also have commercially available kits that can evaluate the microbiome of your dog in different areas, in the intestinal, in the feces, in the skin, in the nares, and so on. So if you want to check the microbiome of your dog, if it is healthy, if it is not healthy, you can send it to this laboratory and you don't pay very much and you can have it done. Okay, now let us see some facts. First of all, there are many microorganisms in and out of your body. Over 40 at 13, so that's a potency. So 40 with 13 zeros behind. So just imagine how many you have inside your body. About 1 or 2 kilograms of your body weight is only bacteria. So this is one extra organ that you carry around, and this organ is doing lots of things that you cannot do, and it is very important. And 90% of the cells in your body are not human cells. If you check in numbers, 90% of the cells in your body are bacterial cells. Only 10% are human. In number, of course, dimension, the bacteria are much smaller. Why is this organ that we carry, that is 1 or 2 kilos of bacteria, why is it so important? Well, if you think about it, the human genome is 99.99% identical between individuals. There's not a lot of variation, and we only have 33,000 genes. And this 1 or 2 kilos of bacteria that you have in your body, they have another over 1 million genes that will produce 1 million proteins. And these proteins are important because they code for functions that our cells do not do. For example, they produce vitamins. For example, they can digest cellulose in, for example, in ruminants. And so there is a cooperation between the human and the microbial genome, so that in fact we are a super organism, we with our microbes. And the microbes are very important because they do influence how we are and what we are. They are very important to stimulate and teach the immune system. And so they are a very important essential component of our immunity, but they are also very important for our metabolism because they produce products that we are not able to produce, enzymes, vitamins, and so on. And I'm going to show you that if you don't have microbes in your body, you are not very healthy. For example, there are these germ-free mice. They are born and they live in completely sterile environment. And so they don't have any bacteria in their body and they don't have any bacteria on their surface. And they have a very bad immune system, very bad metabolism, and very strange behavior. And they die very soon, they don't grow very well, and they are very susceptible to stress and to infections because the immune system has never been taught to recognize and to fight bacteria. Now, regarding metabolism, there is another nice study that has been done, and that is they have taken the feces of an obese rat or mouse, and this feces contain a certain variety of bacteria typical of the obese mouse. They have made a fecal transplant in a normal mouse, and with this bacteria and with a different metabolism, this mouse has become obese. And probably this is the same that also happens in humans. The bacteria that you have in your intestinal microbiome, if you are obese, they probably will determine or influence quite a lot your obesity. So that if you can change your microbiome in your intestinal tract, this can help you quite a lot to decrease your obesity. And in fact, what you eat is going to influence your bacteria. And so if you eat bacteria if you eat too many lipids, you will select for some bacteria that will produce more inflammation, for example. If you eat too many sweets, you will select for bacteria that will cause obesity, and so on. And of course, if they transplant feces of a normal mouse to a normal mouse, the mouse will stay healthy. Okay, so there has been this very big project that was started in the United States. And this project aimed at identifying all the bacteria that were present in the human body. And the results of this process are that there are over 4,000 different bacteria species in the gastrointestinal tract, with Bacteroides, Prevotella, and Lactobacillus predominant. There are 1,200 species on the skin. Just imagine how many we have. Propionibacterium, Staphylococcus, and Corynebacterium predominant, 800 in the oral cavity, and 300 species in the vagina. And when you read these articles on the human microbiome, you see these nice pictures where you have these cakes that are representing the different bacteria and their distribution of amounts in the different body areas. And the different colors are different types of bacteria, and you see how abundant they are in the different body areas. But now I would like to concentrate on the skin bacterial microbiome. So first of all, we have to say that the human skin is not very hospitable for bacteria because our skin is actually cold, acidic, very dry. We don't have a lot of nutrients, only something on the sebum and subpeptides and lipids from the stratum corneum. And we produce quite a lot of sweat. Animals don't produce so much sweat, but we do. And it is salty, and it also has some antibacterial molecules. Not only the salt is actually inhibiting the bacteria proliferation, but also free fatty acids, antimicrobial peptides, ferritin, for example, you may remember, secretory IgA, which are also produced in the sweat. Nonetheless, we have about 1,000 to 1 million bacteria every square centimeter of our skin, with a total of 10 billion bacteria that are covering the whole skin. Just to make an example, this study has found that there were 158 different bacteria species on one palm, only on one hand. And funny enough, the deeper you go in the skin and the more bacteria you see, because in the lower epidermis you have much more water, less salt, much more nutrients, and on the surface the situation is very dry and it's not so easy to live on the surface of your skin. So the species that are more abundant on the human skin are certainly Propionibacterium. This is the bacterium that causes acne in young teenagers. And then we have Corynebacterium and of course Staphylococcus. And in human skin, we see actually different distribution of bacteria depending on the skin type. We have some sides that are more fatty. For example, think at around the nose, behind the ears, on the neck, or on the back, we have more fatty skin. And in this case, we have more Propionibacterium, the one that causes acne, rather than Staphylococcus. But we have other body sites that are dry. And for example, in this case, we have more Proteobacteria and less Propionibacteria or Staphylococcus. So we have different site types, sebaceous, moist, and dry. And depending on the sites, you have different distribution of the bacteria. But the difference is not only depending on the type of the skin in the different body sites. We also have quite a lot difference between individuals, interpersonal variations. For example, these are 4 people, HV1, HV2, 3, and 4. And this is the antecubital fossa, that means the fold of the elbow of the first person and of the second person. And as you see, there are differences between people. This is the back of 4 different people, and you see that this person is really different than the others. So you can have quite a lot of interpersonal variations, depending probably if your skin is more greasy or less greasy, and so on. And also, because we shed so many cells, and the cells we shed are full with bacteria, we actually give an impact of our microbiome to the environment where we live. So we shed between 500 and 3,000 cells per square centimeter every hour. Just imagine how many cells we lose every hour. And many of them have bacteria. And so the room where I am now, it has the impact of my microbiome. But also my 2 girls that are sharing my house, probably they also share a very similar microbacteria to the one that I have, so that we have like a family microbacteria. And this would also include the dog in the family, so that the microbiome of the skin of the human beings and of the animals in the same house tends to become more and more similar. And of course, having a dog in the family adds diversity to the bacteria of the human beings that live with the dog. And this is a good thing, because we want to have a lot of diversity in our microbacteria. And this is probably also one of the reasons why families that have dogs and cats, their children are less allergic, because the immune system has been better educated to react to the environmental allergens. Okay, so we're talking about the immune system and the education of the immune system by the bacteria. In fact, there is a permanent dialogue between the bacteria on the skin and the immune system. And not only the bad bacteria on the skin that will activate the immune system to kill them, but also the resident bacteria on the skin. They are interacting with the immune system, and they are teaching the immune system how to kill the bad bacteria. So it is a continuous dialogue to improve the defense of the skin. In fact, the resident bacteria, good ones, for example, Staphylococcus epidermidis and Propionibacterium, they protect against the invasion of the pathogens, because they are actually educating the T cells of the immune system to respond to bacteria that are pathogenic, that may be similar, and so the immune system will recognize the bad Staphylococcus aureus, for example, because it has already been primed by the resident Staphylococcus epidermidis. And then the bacteria are also able to cooperate with the host antimicrobial peptides to enhance the killing of the pathogenic bacteria. So in fact, this cooperation is very important, and you want to have the good bacteria on your skin in order to be able to kill the bad bacteria. Now, I have talked about the skin of the human beings, but we also have some information on the skin of the dogs, because there are some studies that have studied the skin microbiome in healthy and in allergic dogs. So first of all, dogs are not small humans because we do have differences. We have seen that in humans, the most important phylum is Actinobacteria, Propionobacterium. Then we have Firmicutes, this is Staphylococcus, and as fungi, we have Malassezia, which is really present a lot on human skin. On the other hand, if you look at the dogs, we have Proteobacteria as the most prevalent phylum, and then Firmicutes, the Staphylococcus, and then Actinobacteria like Corynebacterium. And if we look at the fungi, we don't have Malassezia as a frequent member of the microbiome, but we have more environmental fungi like Alternaria and Cladosporium. And I think that this is due to the fact that dogs have a long hair, and they will probably collect the spores and the fungi from the environment, and they're actually passively transporting these fungi that are then recognized with the techniques that amplify the fungal genes. Now, why do we have this difference? Well, probably because the skin is different. It is less lipidic, less greasy in the dog, and we have not so much sweating on the dog, but also because the humans are actually shampooing and bathing and showering nearly every day, and they have different hygiene practices. The dogs walk much closer to the ground. They are much more contaminated with bacteria on the soil, and humans, they don't have a lot of contact with the ground, with the soil, and they are have less environmental exposure. So when you look at the articles that analyze the bacterial microbiome of dogs, you have these pictures that look similar to those of humans, where you have the different body areas, and you have these cakes, and every color is a different bacteria, and the dimension of the cake piece is the relative abundance in that body area. So the most frequent bacteria species in dogs are Propionibacterium acnes, Haemophilus, Corynebacterium, and Staphylococcus epidermidis, not Staphylococcus pseudointermedius. I'm talking about bacteria that are normal resident on the skin of dogs. And we have some differences depending on the area, the axilla, the dorsal neck. We may also have some differences depending on the season. But in fact, we don't have the clear distinction of differences in populations depending on the skin type or body site that we have in humans. We don't have that. The effect of the body site on the richness only accounts for 20% and not so much as with humans. We only have some moist body sites that are the lips, the nostrils, and the conjunctiva, where we know that the richness, the diversity of the bacteria is much smaller. So this is the richness, the diversity, the number of different species. And we see here that in moist body sites, the number of species each is smaller. It's like that when you have a humid body site, then this humidity will select only for some bacteria that are able to survive and some others are not. While in the rest of the body, you have a large variety of different bacteria. Also, the individual dog accounts for about 50% of the richness. That means that we have many differences between dog and dog. And also, we don't have a great variety depending on the breeds. Breeds account only for 9% of variability. Much more is important the individual differences rather than the breed differences. And I'm going to show you here. This is one breed. These are the French Bulldogs. These are the German Shepherds, and these are the West Highland White Terriers. And each one of these bar is a different site. And this is one dog, this is another dog, and this is a third dog. And you see that we have much more difference between one dog and the other dog in the same breed, rather than, for example, differences between breeds. They all look sort of similar, so there is not so much of a difference between breeds, rather differences between the single dogs are much more important. Another observation that was done is that healthy dogs that are not pruritic, don't have skin lesions, don't have infections, don't have seborrhea, nothing, they just have a very strong odor. In fact, they do have a dysbiosis. And in these dogs, we see a lower microbial diversity. We see a higher abundance of malassezia that probably accounts for part of the bad odor, and more staphylococci, in particular Staphylococcus pseudointermedius. It is like in these dogs, because there is less diversity, we have more staphylococci, which is typical of the dysbiosis in allergic dermatitis. It could be a prodromic phase of maybe developing a bacterial infection later on. So let us see in animals that have atopic dermatitis, what happens in the skin. Well, we do have a lower number of bacteria species. And this is actually a dysbiosis, because we should have a high number of different bacteria species to have a normal skin. If this is not the case, this is not normal, and we can call it dysbiosis. So for example, the blue line is healthy animals, and this is the number of bacteria species that we have observed. And the red line is the allergic dogs. And you see that the blue line has always higher amounts of bacteria species that have been observed if compared to the red lines. So this is typical also in humans, that when you have a atopic skin, you have much less different species and much more Staphylococcus. And this has also been proven in this article. Here we have 14 dogs with atopic dermatitis, and they had actually lesions. They had a disease flare. And in these lesions with disease flare, you see a much lower diversity index in atopic dogs, the red line, if you compare it to the diversity index in the control dogs, that is much higher. This is the pinna, the axilla, this is the pinna, this is the groin. In every skin site, the diversity of the healthy dogs is always higher rather than the diversity of the atopic dogs. This is not the case in mucosa. And this is the similar, showing the similar concept. This is the axilla of these dogs, atopic dogs and control dogs. The pinna, atopic dogs and control dogs, and the groin, atopic and control. And you see that the control dogs always have many more colors rather than the atopic dog. And if you have many colors, this means you have a large diversity. While in the atopic dogs, the rose, the pink color is always larger, and the other colors are much smaller. And this means that the majority of the bacteria now is Staphylococcus and is not all the other nice variety of bacteria. And we have seen before that the number of the staphylococci on the skin correlates with the severity of the lesion, erythema and pruritus in the atopic skin. And this is when the animals have an acute flare, but when you treat your atopic dermatitis and you restore the skin barrier, you restore the inflammation, you restore the erythema, it goes back to normal. You also restore your bacterial diversity on the skin. So this is also correlated. Here you have the animals that have atopic dermatitis, the red ones. The first visit, when they have the acute symptoms, the diversity is lower than the healthy control. When these animals are treated, when they are looking with no pruritus, no erythema, no lesions because they have been treated, here you see that also the bacterial diversity is increased and is not the same diversity as the healthy controls. And you see the same concept here. This is the healthy animals, lots of colors. This is the atopic dermatitis dogs, a lot of pink of the Staphylococcus, not so many other bacteria. When you treat these animals, you see that the Staphylococcus decreases and you see many more colors, many more other bacteria. This is the diversity that is improving quite a lot. And this diversity is really important because, as we have seen before, it correlates with the skin lesion severity. And this is why I was speaking before of the importance of restoring the diversity on the skin and helping the immune system to kill the bad bacteria on the skin so that the good bacteria can come back. And as I was saying before, link skin is able to do this because these lactobacilli, they are going to influence the immune system to normalize the bacteria population on the surface. But not only, there are also some algae extracts that will favor the growth of the normal microbial flora because they are nutrients. And you also have some vegetable polyphenols that will decrease the inflammation and are antioxidant. And if you decrease the inflammation, you will go back to an increased bacterial diversity, which is so important. Now, there is a study that has been performed by Domenico Santoro, who is a person that works in dermatologist in atopic dermatitis in the University of Florida. And he has taken 6 atopic dogs that had lesion on the ventral areas, and he sprayed these dogs with lesions on the ventral areas every day for 28 days with lean skin. And he saw a significant decrease of the lesions, and 67% of the dogs, 2/3 of the dogs, reached lesional scores that were very low, below 10. So you see, these are the lesions at the beginning of the study, and within 2 weeks, the lesions would decrease very much, and they will persist until the end of the study. So this can be a nice alternative, innovative way of treating atopic dermatitis topically. Okay. Now we have talked about the skin microbiome, but we have a gut-skin connection. We have the intestinal microbiome that is very important to maintain the health of our skin. And I show you why. Well, you have to know that 70% of all immune cells are in the gut, and 90% of all antibody-producing cells are in the gut. So here you have the intestinal lumen, and you have lots of bacteria and also lots of products that are produced by this bacteria. This is the intestinal wall, and behind the intestinal wall, there is this very potent immune system. And there is a lot of interaction between bacteria and their products with the cells of the immune system. And this interaction can actually cause different results. Here you have, look at how many bacteria you have in products and how many different cells that could come in contact with these immune cells. And you can imagine of the many results of these interactions. It can be that the immune system is instructed to react in a healthy way. It can be that the immune system by the bad bacteria is instructed to react in a bad way, for example, in an allergic way. And if you have the bad bacteria in your gut, it may be that the allergy that is the consequence of the bad bacteria will appear in the skin. So in fact, an intestinal dysbiosis can disturb the efficiency of the immune system and can contribute to diseases even in the skin. And this has been shown by different studies in humans. For example, they have found differences in fecal microbiota in people that were atopic and people that were healthy. And the differences are so important that if they look for these differences in the fecal microbiota in children, they can predict which children will develop atopic dermatitis and which children will stay healthy. So in some case, somehow, if you could improve this fecal microbiota, if you could correct this microbiosis, you could change the course of the disease and change the fact that these people will become allergic. Of course, antibiotics will kill the bacteria, will cause some disturbance in the bacteria in the gut, and we know that giving antibiotics is a risk factor for developing atopic dermatitis in the future. On the other hand, there are some studies in human beings that if you give good bacteria like Lactobacilli, bifidobacteria in patients with atopic dermatitis, this will improve the microbiome, this will teach the immune system in the intestinal tract to react in the good way, and so it will improve the clinical signs of atopic dermatitis, also by decreasing Clostridia that are not very good. Now, thinking at this concept of improving the microbiome and thus decreasing the allergy, there is one study that has investigated this in dogs. And this was done by Rosanna Marcella in the United States. And she has done a very elegant study. So she has taken a couple of dogs that were at high predisposition for allergy, a male and a female, and she mated these dogs twice. The first time that the dogs were mated, the offspring, the puppies, were not treated. And the second time that the same parents were mated, the puppies of the second time, they were treated. Actually, the mother the second time was given Lactobacillus rhamnosus during the pregnancy and during lactation, and this same Lactobacillus was given to the puppies between 15 days of life and 6 months of age. And then these dogs were stimulated, sensitized with house mites, and the dogs of the second time, the dogs that were treated, compared to the dogs that were not treated, they had a decreased allergen-specific IgA production, decreased skin test reactivity to house mites, and decreased incidence and severity of clinical signs of atopic dermatitis. This was done when the dogs were 6 months of age. Then they were not treated anymore with Lactobacillus, but they were sensitized after 2 years with another allergen, and still they showed decreased clinical scores and IL-10 production. So this imprint that they have received during weaning and in the first 6 months of age, that this lactobacilli gave to the intestinal immune system, this produced dogs that were less prone to developing allergy for many, many years afterwards for the rest of their lives. And so with this in mind, there are some products that come now on the market that are actually, they actually want to improve your intestinal microbiota in order to improve the skin disease. And this is one of these products, Lean Skin Tablets. It contains Lactobacillus rhamnosus and reuteri, also essential fatty acids and defensins. You see, these are good for, these are antimicrobial peptides. And by improving the microbiome, by stimulating the immune system in the guts, Lean Skin wants to improve the health and appearance of the skin. Now, you may ask, what is Lactobacillus reuteri? And it is a normal component of the intestinal microbiota in cats and dogs, helps normalize the intestinal microbiome, inhibits development of pathogenic microorganisms by decreasing their adhesion and increasing mucosal EGA. You see, it works on several aspects of the interaction between the immune system and the bacteria. Okay, now I would like to show you the last few slides because we have talked about a gut-skin connection, but this is not all, because we also have a gut-brain connection. That means that the gut microbiota are able to modulate what happens in our brain: stress, pain perception, neurotransmission. So now we know that the microbacteria that we have in our intestinal tract, they are actually able to modulate our mood. And so if we eat well and have a good microbiome, we are also of a good mood. Now, this has been proven in mice. For example, Citrobacter produces substances that cause a vagal activation, and this will lead to anxiety. Or Bifidobacterium, which is good, you find this in some probiotics and yogurt. It will increase your intelligence, increased cognitive processing. Or even you can change the personality of a mouse by making a fecal transfer. For example, they have taken some very timid mice and they have put some feces of very courageous, bold mice, and these timid mice have become bold because it has changed their personality in their brain. Also, we know that in germ-free mice, there is a very bad formation of nerves. They have altered branching, altered myelination. So bacteria help you develop your neuron system, your intelligence, decrease neurogenesis, decrease serotonin, and these poor mice are depressed. And in fact, we have seen that the different bacteria, they can produce a variety of different molecules from serotonin, histamine, phenylalanine, dopamine, melatonin, GABA, and each one of these molecules can have a different effect on your mood, like mood control, anxiolytic, and some of them give hypertension and so on. And now, in the last few years, there are some studies that have determined that dysbiosis in the gut may be associated with neurodegenerative or neurodevelopmental diseases, such as Parkinson's disease, for example, autism, schizophrenia, and neuroinflammation. So now what is happening in the human medicine is that they are trying to produce some cocktails of good bacteria, and they are trying to give it to people that have neurodegenerative diseases like Alzheimer's disease. And it seems that this is going to help people with Alzheimer and with Parkinson, because there may be some benefit even in the brains if you have a good microbiome in your guts. And I have found a veterinary article that is also quite astonishing and interesting, where it is proven that the gut microbiome in dogs will correlate with the aggressivity of the dogs. So depending on the gut microbiome, dogs will be more aggressive or less aggressive, which is also something that I think will be part of studying in the future, how the gut can actually influence your mood and your personality. Okay, so this is the last slide of this lecture. The key messages regarding the microbiome are that, first of all, there are many more bacteria in our body and on our body than we ever could imagine, and they are very, very important for our health. On the other side, if we have alterations of the microbiome like dysbiosis, we also have disease, and this disease can be also of the skin. And I think this is just the beginning of a very nice and fascinating travel. We are going to see many more data, many more studies in the future, and hopefully many more products to help us in a natural way to solve our dermatological diseases. Okay, Thank you very much for your attention, and I am ready for some questions if there are any.
I would like to thank you, Chiara, for an interesting and I believe very useful presentations. And please ask the questions. Take your opportunity to talk to Chiara.
Yeah, I can see in the chat there is a question. What is protist? Protista proteozoa, like for example Leishmania. These are unicellular cells that have a nucleus. So they're not bacteria because bacteria don't have a nucleus, but unicellular cells with a nucleus. Okay, any other question?
Don't be shy, ask.
I hope that they found the information on microbiome interesting, because I think it is really a nice new field of medicine that will give us a lot of information in the future, and it's really fascinating.
Yeah, I'm the same opinion. And I believe that when the participant will digest all the information, that the questions will come afterwards.
Was this too quick? Was I too quick when I spoke?
No. There is somebody talking, but now mute the microphone. Chat.
Okay, at least Petra thought it was an interesting lecture. Yeah. Thank you, Petra. One.
No, I think more that they are shy, but don't be shy, ask whatever you need to know.
Hi, Sretanka. Okay, so what is our host saying from Slovenia?
Mojca or Alenka, are you there?
Hello, Katerina. I'm here.
Alenka, hi. So.
Hi. Very good.
Hi, Mojca is also here.
Hi.
I'm sorry you didn't. The situation is like this, that you couldn't come to Slovenia in person. There was a lot of interest before the pandemic began, so we had to cancel the event.
But there is a
good. It's a pity.
Yeah, but we have a good news that there is a promise from Mr. Falanga that Chiara, when everything will be okay in autumn, that we can organize a live seminar, not a webinar. So we should cross the fingers that it will happen.
We cross the fingers.
Yeah. Of course, then we need to find another topic, Chiara.
Okay.
So
I don't think we have to find another topic.
I have lots of topics. It's not the problem.
Okay, great.
I have, yes, not a problem. And Maya Holicek says that we should all use less systemic antibiotics, and I agree 100%. Anything we can find to avoid systemic antibiotics and substitute them, we should use. And I have to be sincere, I was using a lot of antibiotics, and in the last 2 or 3 years, I nearly ever use antibiotics. It's very rare that I give antibiotics now. It is possible to change the way of working with the same results. It is really possible. I didn't believe it in the beginning when everybody said, don't use antibiotics. I said, this is not possible. But now I know it is possible to only use very few antibiotics. It is possible, yes. Now I understand the way link skin works. That is good. I'm happy, Petra, that you understand. It took me a while too to understand, because ICF always does very innovative things, and sometimes they are so innovative that we don't understand, but with time we understand that they are really, really intelligent products.
Thank you, Chiara.
Okay.
So if there are no more
Good.
questions,
Then good night.
I only want to say that if
If there
the
are
questions
no
will
other
come
questions,
later on, please send them to Alenka or to Mojca, to Iris, and then I can forward to Chiara and she will reply to you. Okay.
Yeah.
yes.
So thank you for all for
Okay.
attention.
Good night
Thank you,
then.
Chiara, very much.
Bye-bye.
And hope to
Bye-bye.
see you
Bye.
alive. Bye-bye.
Bye.
Bye-bye.
Bye.
Goodbye. Thank you.
Bye. Bye-bye. Bye-bye.
Bye.